test exam. I know 3 weeks before the exam, but it wouldn't be a bad idea to start studying. It'll be straightforward. It'll be right off the study guide, right off of my notes. I don't do anything tricky. That's not my goal to do it tricky or anything else. And you know, you can go down and see everything we've talked about so far is right on here. And there'll be questions about all these different topics. And we have a lot to go on this. So we haven't gotten to some of these topics yet. But and when you're studying, if you have questions, something doesn't make sense in my lecture, or you're not quite sure about something, email me. That's fine. I don't mind. If you want to try to write out an answer, and you know you want to make sure it's a good answer, you can email me. I'll look at it and I'll critique it. So that's I don't mind doing that. So don't hesitate to do that. But anyway, we have 3 weeks, as I said. I'll try. I haven't found an open room yet, but I'll try to do a study session the Friday before that, or the Monday. I don't know. Monday's tough because there's not much time for me. But the Friday I have time, so maybe the Friday before, if we could do a study session where I can answer just general questions that are still confusing you that haven't been cleared up. Any other questions, you guys? Any questions about what? Yeah.
Can you answer another study guide for this one?
Yes. So you're going to study twice as hard too, yeah, right? Good.
Oh,
So,
okay.
want more? No. You think I'd forget it? Anyway, okay, so let's. I got to find some more colors because I like all the. Okay, so one thing before we go any further, just an announcement just about what's happening in the biology department. How many of you are thinking of doing the nursing, pre-nursing year? Okay, so you're going to need to take anatomy, physiology, and micro. Okay, starting 1 year from now, next spring. So it's a year from now. Chemistry will be required for micro, and anatomy will be required for micro. In other words, if you take anatomy, if you take physio next semester, chemistry isn't required. But if you take it the following semester, I might be teaching it next semester, I don't know yet, that's a possibility. But if you take it the following semester, anatomy will be required and chemistry will be required. Now, if you're thinking about going to nursing here at CR or anywhere else, I highly recommend you take chemistry, Chem 2 at the very minimum. The more chemistry you take, the more things make sense, you know. But I know C is the dreaded, chemistry is the dreaded C word, and people are afraid of chemistry. And yes, I think chemistry is not simple. It's tricky, but you can understand it. I want you to be aware of that. Now, I don't know what this is going to do to my physio enrollment for the next year, so might cut them back because people haven't taken the chemistry and my anatomy. But I recommend that you start planning whatever you're going to do, keeping in mind that chemistry will be required a year from now, as will anatomy be required for physio. Question?
So if you mean it's required for physio and not for micro?
Not for micro and not for anatomy. So chemistry is only going to be required for physio. But if you haven't had physio yet and you want to get into the...
So if we take that in the fall...
You're correct. Yes, that I am saying that, which means everyone's going to probably try to take the. Oh, everyone who's afraid of chemistry is probably going to try to take it in the fall. Yes, which may not be possible. I mean, we probably won't be able to fit everybody in. But just plan, you know, and if you can't get in, then see if you can get into chemistry in the fall and take it in the spring or something like that. You know. But also anatomy. You want to take anatomy first too. Go ahead. No, not human biology. But you wouldn't need human biology to take it. Well, I'll talk to you. If you want to talk about human biology, I'll talk to you. Yeah.
Does physio run in summer at all?
I don't think they'll do it this summer. My guess is not. Sometimes we do. I have taught it this summer, but I don't think it. I'd like to teach it in the summer, but I doubt that it's going to be offered this summer.
No, it's just one semester.
Yes, just the Chem 2. Chem 2, not Chem 1A.
No, not Chem 1A. It's Chem 2, the simpler Chem.
Yeah. I don't say simpler Chem. Let's just say it's reduced Chem.
I thought it was like an introductory Chem before Chem 1A.
No. Chem 100 is the intro for Chem 1A if you haven't had chemistry before. Chem 1A and 1B are the majors chemistry class. If you're going to go on and do science and stuff like that, physical science or biology, you take Chem 1AB. But if you're going to do like health occupations, then you do Chem 2 and sometimes Chem 3, depends upon the school you're going to. When that other school up the way used to have a nursing program, I mean, the fact that Humboldt dropped their nursing program, I think, is the stupidest thing ever. And you can quote me on it, but whatever. But you know, since it's a bachelor's program, you had to take Chem 2 and Chem 3. In the old days, we required chemistry for biology, or for nursing, but it was just Chem 2. You didn't have to take the Chem 3. But if you're going to do a bachelor's program, usually they want the Chem 2 and Chem 3. Because Chem 2 is an intro to chemistry, and then Chem 3 is the intro to organic and biochemistry. So, anyway, so that's the deal. Okay, so any other questions? So that was an announcement, just so you start preparing. It's kind of important that if you're going to take those pre-nursing classes, think about what your schedule is going to be.
Generally during the summer, they don't offer any of the biology classes.
Chem 2 is going to be offered this summer. The plan is that Chem 2 will be offered this summer, which would be a good time to take it, so you're only taking chemistry. It doesn't make it easier, but then you don't have all these other things to worry about, you know. I mean, presumably, you still have life to worry about, I suppose. But when I teach anatomy, by the way, I haven't taught it for a number since I retired. And of the classes I taught, it's the most demanding class to teach, and it's the most demanding class to take. And I think it's one of the two most demanding classes on campus. But when I would teach
The other one being
probably so. But when I teach anatomy, I would say to my students, the first day of class, you know, by the end of this semester, you'll hate me, your partners will hate me, your pets will hate me, your kids will hate me. Everyone's going to hate me because you're spending all your time studying anatomy. So you guys who are planning on doing nursing, my recommendation, it's not an absolute here, but my recommendation is you take anatomy on a semester without any other sciences, and not with a lot of other demanding classes, because you're going to be busy. And if you're going to double up on classes, take physio and micro together or something like that. So anyway, that's just my recommendation. You have to do what works for you guys. Anyway, questions? Any other questions?
I was just going to make an amount. This actually turned out to work. I was going to start recording all of them and post.
Is it working?
Yeah, it worked out really good. Actually, I can hear you really well. So if anybody wants, probably the next class meeting, or I'll send out an email. All these lectures are going to be available online. I'll figure it. I'll put them up on YouTube or something.
Oh! I have, I did record all my lectures for physio, and people wanted to put them up on YouTube, and I thought, oh God, I'll probably get all these obscene comments.
You can shut down the comments.
Oh, can you?
Yes.
Okay, so if you put them up, don't let them comment.
I'll do that.
And the dorky clothes he's wearing and the stupid haircut. I don't want to see that kind of stuff. He's so old he can still talk. I don't need to see these things. Anyway. All right. So so we got the midterm. We talked about that. Okay. I think we can start back on cells. Cells. We're going to talk about cells. Remember the two kinds of cells? Let's just write that up again, because I'm going to be talking about that again. I mean, I want to keep this in your mind. What are the two kinds? Does anyone remember?
Yeah, eukaryotic and prokaryotic.
And eukaryotic. And we'll see some of the differences on these guys today. And what are the only organisms that are prokaryotic? Bacteria. 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 And the blue-green bacteria. Did I step on any toes last time about saying my things about the blue-green algae? Does anyone take it here? I'm just curious if anyone takes it. Or sells it. Okay. Well, yeah.
Why do they call blue-green bacteria blue-green algae?
Because it grows with algae.
And they just haven't learned that it's not algae. Because algae, true algae, algae are real things, but they're eukaryotic. So it's just, and actually, by the way, nowadays, this is just fairly recent, maybe the last 10 years or so, they in their classification of all living things, they used to always start with the kingdom, the animal kingdom and the plant kingdom and the fungal kingdom, but now they actually start at a higher level, and this is the domain, and there's one domain of the prokaryota and one domain of the eukaryota, so they actually separate the two domains, and then in the eukaryotes are all the animals, plants, and algae, fungi, and all that sort of stuff. So it's just, which is a good thing. I mean, I don't think that's a bad thing. I think that's a good thing because it does help differentiate the fact that there are two fundamental types of cells. What we're going to see, though, today, so let's see, everything else. That's not right. Animal cells. We did this last time, I know. Plant cells, fungi. I should tell you my little joke. Say protozoa, algae, etc. You know, a few other things that fit there. But that's most everything. Okay, I'll tell you my little joke. And by the way, I know how corny these things are. I'm totally aware of how corny my jokes are. It's okay to just go, ugh. You're not going to grow. Okay, so yes, so Sally Strawberry and let's see, let me have to think of something to see. And Betty Broccoli. Okay, Sally Strawberry and Betty Broccoli were in school together, and the prom was coming up, and they were saying, hey, you know, check, have you been invited to prom yet? And no, I haven't been invited to prom. How do you know? So anyway, so they keep talking for several weeks. These guys, Sally Strawberry and Betty Broccoli, so you know, saying they're going to go to prom. Finally, one of them, Sally Strawberry says, "Oh, I got invited to the prom. Oh, great." So then Betty Broccoli says, "I haven't been invited yet." So she said, "Well, actually, that's not true." She said, "I did get invited by Robert Radish." Well, then why are you going to go to the prom with Robert? No, I don't want to go to the prom with Robert. He's not fun to be around, you know. Oh, okay, so. You know, the next day, Sally Strawberry says to Betty Broccoli, "So did you get invited by anyone else now?" And she said, "Yeah, I got invited by Walter Watermelon." "Why are you going to go to the? No, I'm not going to go to the dance with Walter. He's boring. I don't like being around him." So finally, she said, "Hey, I'm going to go to the prom." Says Betty Broccoli, and Sally says, "Well, who are you going to go to the prom with?" She says, "Mortimer Mushroom." He says, "You're going to prom with Mortimer Mushroom?" "Yeah, he's a fun guy." Oh, I told you it was Anyway, that's going to be on YouTube, I suppose.
Yeah.
Oh, great.
And my roommate has a shirt depicting that joke.
Oh, did he really?
With the mushroom says, "I'm a fun guy."
I'm a fun guy.
Yeah.
By the way, just for interest sake, you don't need to know this, but this is just sort of, since we're on this sort of off the topic, properly is fungi.
Yeah.
You know, the singular is fungus, but the plural is fungi. Yeah. And what's interesting to me, and I don't teach English, but I do love words. It goes from the hard G to the soft G, from the singular to the plural. Fungus, fungi. Okay. Same thing happens for alga. Alga is a singular. This is singular. You're not tested on this, by the way. And this is plural. That's alga and algae. Alga and algae. Again, why it changes from the soft G to the hard G, or vice versa, actually. I believe from singular to plural, I don't know. If you take anatomy, they're in the stomach. The stomach, if you open up the stomach, there are a whole bunch of these little folds, and they're called ruga. I mean, each one's a ruga, but the plural is rugi. I always tease my students when they forget the name of that. I always tell them, "Gosh, how can you forget Rougie? Because there's a whole celebration, Rougie on the river every summer."
Ha!
Oh, God. See what I mean out there? We got to get back to this. All right, so what are we talking about? Oh, yes, we're going to talk about cell membranes. So if I just do a simple picture, you know, I told you here's the simple picture. God, you got me wound up now. I didn't even have any coffee. Well, I did this morning. Okay, so I'm going to look at a little tiny portion of the cell membrane. And I said cell membranes over here. I said cell membranes are made of phospholipid bilayer, and proteins. And the way we were depicting phospholipids, I get some fresh markers. I got a correction on the marker on the roof. Let's see if this is better. The way I was depicting, I mean, the way we usually depict, I showed you this last time. The typical way we do a phospholipid, you know, in terms of a picture, yeah, we have this little circle with the, you know, which is the phosphate head. Remember the phosphate? So that's a phosphorus with
A little lipid
oxygen
tail.
and hydrogens attached to it. That's the phosphate head, as they call it. And then we'll have the little fatty acid tails. And if you remember from my, you know, the drawing showing the glycerol, the 3-carbon sugar with the 2 fatty acids attached to the sugars, and 1 sugar had the fatty acid. I mean, the phosphate head. That's a better chemical description. This is a nice, simple little drawing. But the tricky thing to remember, it's not really tricky, I mean, the main thing to remember is the phosphate head is hydrophilic. It likes water. Because it's charged. It's easy, it's nice, it loves, you know, ions love water, water loves ions. But these fatty acid tails, these are fatty things, these are hydrophobic. These are hydrophobic, they don't like water. They like to be as far away from water as they can get. And, you know, in the lab, you did that oil and water experiment last week, right? And you know, and the same thing if you had an oil and vinegar salad dressing. You know, the oil and vinegar don't like each other. They separate. There is that one little area where they're sort of next to each other, but they're not happy. Neither one is happy that they're next to each other. And the same thing with these fatty acid tails. They don't like water. They like lipids. They like fats. And if you try to shove those little lipids into water, they don't like, the water doesn't like that, and they don't like it. So it turns out that in a nice, easy way, these phospholipids get together in this bilayer, and everybody's happy. Okay, now that's a different happiness from the having the right number of electrons. Yeah. They're happy again because they're the things that like water are near water, the things that hate water are away from water. And so the bilayer then has a whole bunch of these guys, and I just started by drawing a simple little picture last time. And I said I'm drawing it like this, but they're much more crowded together, and of course it's all 3-dimensional. I'm just showing in just 1 dimension, I guess 2 dimensions, really, what it looks like. But of course, we keep going this way, keep going this way. So I mean, it's just very oversimplified. But here would be one part of the bilayer, the phospholipid bilayer. And of course, as I said, they're much closer together. There's not all the space between them that I'm making. I'm just doing it so you can see it. And then this one has the little tails here. But notice that the tails are all next to each other. They're the guys that hate water, but they like each other because they're not near water. So things that are hydrophobic like each other, but they don't like water, and water likes hydrophilic things and doesn't like the hydrophobic. So they can form this bilayer. Remember what I said was there's always water on the outside, and there's always water on the inside of cells in general. Essentially all cells. Pretty rare to find cells with water on the outside. They have to have some way to keep the moisture in. But anyway, so and so this would be the outside here in my drawing, since I just took a little piece right there, and here's the inside. And so the part that likes water, the hydrophilic part, the phosphate heads, on the outside they're exposed to water, on the inside they're exposed to water. But these tails are right next to each other, and they're away from the water. Water is being excluded from where they are, and they're happy being together, and that they're actually held together. There's actually a term for that, and I'm not going to ask this of you, but I'll just mention that talks about hydrophobic bonds. And all that really means is that hydrophobic molecules will tend to stick together and try to exclude water. And what would happen if you tried to open up this membrane? Water would try to come in here, but water doesn't want to go in there because it's hydrophobic inside. And the fatty acid tails don't want water there, and so water doesn't want to go there. So it'd rather stay together, because it's nice and stable, and everyone's happy. You know? Not to say that they're tough. cell membranes are tough. They can be broken fairly easily, but relatively speaking, they will stay together as an intact cell membrane. Now so the bilayer means that, hey, here's a layer right here, here's a layer right here, 2 layers of phospholipids. All right. Now that's half the membrane. That's roughly 50% of the membrane. So roughly 50% is the phospholipid bilayer, and 50% is the protein. And remember, I mentioned that books make a real big deal out of the phospholipid bilayer and only a small deal out of the proteins. And I want to make a big deal about both, maybe a bigger deal about the proteins, because it's the proteins that are part of the cell membrane that are absolutely critical for the function of the cell. In other words, the phospholipid bilayer is pretty similar in all cells. Pretty similar. There are differences. I mean, even bacteria have these phospholipid bilayers and proteins. I mean, there's still all these membranes are made the same way. And in the old days, they used to think, and then in the old days, it's only like 50 years ago, they used to think that pretty much all cell membranes were alike. Well, they're more or less alike with respect to the phospholipid bilayer. But what makes them different is the proteins. And the proteins, what did I say last time about proteins? They're the worker molecules. Okay? So that they're rather important because they're the worker molecules. And what makes one cell different from another is the kinds of proteins that are in the cell membrane, in or on the cell membrane, because that's going to determine what that cell does, to a great extent. Or at least what gets inside the cell and what gets out. Let's put it that way. What gets inside and what gets out is pretty important for the cell. Let's just stop for a second before I get to the proteins and just say something about it. The cell membrane is a barrier. I mentioned that. I used the term last time. It's a barrier, and I said it is. We're going to see an active barrier, not like your passive wall, for example, that's in this room. It's an active barrier, but it's a barrier. Now, what about things that can go through the, this phospholipid bilayer? We're not talking about proteins right now. We're going to talk about those in a minute. Just the phospholipid bilayer. What can go through it? Not much. Not much.
Lipids.
Because of all this whole area here, this hydrophobic. Let me just write this down. It's not right? Yeah. We'll just use this stuff. How about greatness here? Hey, that's nice. So that's the phospholipid bilayer. And I don't want to have to start thinking about YouTube on this. I've got to be careful what I say. I don't want to make too many terrible remarks about politics or anything. Oh, I'll do that anyway.
Oh, I was going to say.
But anyway, so what can get through that? Well, hydrophilic molecules do not go through the cell membrane easily at all. To go through the phospholipid bilayer. I'm changing. I said cell membrane first, but I'm saying, we're only talking about the phospholipid bilayer. We're going to come back to other things. But things that don't get through are things that are hydrophilic, and things that dissolve in water, like amino acids and sugars and things like that. They do not go through the cell membrane easily because of that hydrophobic area. This little area here is hydrophobic, right? But what can get through are lipid molecules.
Yes.
Lipids can get through this.
Oh, yeah.
Lipids can penetrate phospholipid bilayer. Because, lipids like that hydrophobic area. The hydrophobic area likes lipids. If you use a steroid cream, for example, cortisone cream, that's a steroid cream, that can be absorbed easily through cell membrane because it is a steroid. But, if you put a protein on there like collagen, the collagen isn't good here. If you want to get collagen, they don't get the collagen in the lips.
Yeah,
anyway, that doesn't, you can't get collagen to go through a cell membrane. First of all, it's a huge molecule, and second, it's a protein. And, that's not going to penetrate the cell. Okay, so, all right, so lipids can get through, but otherwise, here's the weird part, though. Water can't. Now, you say water, water likes water. How can it get through? They're not absolutely certain, but they're pretty sure the main reason is just because it's so tiny. Water is so much smaller than things like, say, amino acids or sugars or something like that. It's so tiny, so yes, it can get through. But that's not an absolute. Lipids get thinner by there, and usually, because it's so tiny, it's so small. But that's not an absolute, and if you do take physio, and you take my physio class, I'll actually talk about the kidneys at one point, and there are various cells in the kidneys, there's thousands of tubes, millions of tubes that make up the kidneys. But there are parts of the tubules that freely allow water to go through, and other parts that do not allow water to go through. And there are some parts of the kidney tubules that can change whether water can get through or not, depending upon hormones secreted from posterior pituitary. So that's why I put usually water can penetrate. And again, you wouldn't expect it because it is hydrophobic, and obviously water is hydrophilic. It's water, it loves other water molecules. But it can get through, and presumably it is because it's so small. Okay, does that make sense? All right, now what about these proteins? There are all kinds of proteins. And, in the old days when they first thought about these things, they just thought that all the proteins were, pretty much the same. There's just a whole bunch of proteins. And pretty much the proteins cover the cell membrane, both in the internal part of the membrane and the external part. They pretty much cover it. But starting around the 1960s, they started realizing that different cells had different combinations of proteins. They also started realizing that some of those proteins in the cell membranes are only on the surface of the membrane. Some go through, some go partly through, some go all the way through. And the other thing they found out is that in many cases, the proteins that are here, I haven't put any proteins in here, but they can move. They can move through in that membrane. Okay, they can move within the membrane. That's not true of all proteins, but many of the proteins can move. I'll give you the term for this, the model, but I'm not going to discuss the model just yet. But this is the so-called fluid mosaic model of the membrane. I'll come back to this in a minute, but I want to put it up here, fluid mosaic model of the membrane. I have to be careful because I tend to say cell membrane, but notice if you looked at my outline here, we're going to be talking about membrane-bound organelles. These are membranous structures inside cells. And in other words, there's a membrane around those, and the membrane is very similar to a cell membrane. Okay, different sets of proteins, but it's still going to be a phospholipid bilayer. So that's why I said fluid mosaic model of the membrane, because I'm trying to make it more general, not just talking about cell membrane, but even internal. Anyway, so let's go with the proteins now. I agree. I was agreeing with proteins. So, when they started looking at these proteins, and by the way, these things are tiny. When you look at a microscope and you see what you think is a cell membrane, it's really like a shadow. Cell membranes are so tiny, so thin, you can only see them with an electron microscope. And you say, hey, I can see this membrane like in a plant cell, and it's shrunk. We'll do an experiment where we're using salt solutions and we'll kind of pull away from the cell wall. You're really seeing what amounts to a shadow of the membrane. You can't really see the membrane. They are so tiny. So learning about these things, trying to figure this out and all that, it's not so easy. And they had to do all kinds of fancy stuff with the electron microscope and shade the proteins. But anyway, so I'm going to give you a simplified picture now of that. Now put proteins in green. And so I'll say, okay, hey, here's a protein here. And maybe here's another protein right here, but part of it sticks in here like this. And maybe here's a protein, here's a protein that's just over here, maybe. And then here's one that sticks all the way through, like this. And here's another one that just goes through partly. Yeah. So this would be protein. These are proteins in green. If the protein is only, let's see, what do they call it? Integral. They call it. Have several terms. Let me give you the right term. I don't always use these terms, so I don't know. I might say it the wrong way if I say it the right way. But one of them is integral proteins, and the other, or sometimes they say external proteins.
Peripheral?
What's the term?
Peripheral membrane?
Say it one another.
Peripheral?
Peripheral, yeah. Did I put the terms on the thing?
No,
Yeah, integral and peripheral. That's what the term. Those terms. If I use them, That's what I use. Peripheral, yeah, peripheral. No, not peripheral. Peripheral and integral, yeah. So if the protein is only on the outside or inside, but doesn't go through the membrane, then you just call it a peripheral protein. This is not a big. The peripheral and the integral thing is not a big deal for me. But you should at least know that. So this would be a peripheral protein. The way I drew it here, because it is on the surface of the membrane, but doesn't go into the membrane, not into it. In other words, it's not going into the hydrophobic bond. Whereas, say one of this kind up here or this kind here, they would be called integral proteins.
Ones that go all the way through?
Well, partly through or even all the way through. I mean, you can be any of them. So that'd be an integral protein. I don't know if they have a specific word for ones that go partway through. It's not critical for my purposes. Integral proteins. Part of the protein. Or the protein goes into the and maybe all the way through, as I showed in as I said, now this integral thing versus protein, it's not a real big deal to me, other than that you conceive in your mind that there are various proteins associated with the membrane. And these proteins are going to have functions, and I'm going to talk about some of the functions. And they're critical. Remember, proteins are the worker molecules, right? Mm-hmm. So what proteins are here happens to be critical. Now here's where books tend to shortchange this model, this picture. They'll kind of draw a picture the way I've drawn. And they'll show the nice drawing of the phospholipids, and they'll show these nice proteins in summer, integral and summer peripheral and all that. And that will look fine. But here's the thing I want you to have in your mind, just somewhere in your mind. It's not on a test or anything, but just somewhere to have in your mind that the surface is covered with proteins. The inner surface and the outer surface. That's the inner surface in my drawing, the outer surface, but whatever. They're covered with proteins. Many of them go in, many of them don't. But the point is, my objection to a lot of the drawings is they just sort of, here's a protein here, here's a protein there, oh, here's another protein. In reality, filled with proteins. And keep in mind that roughly 50% of the membrane is made up of proteins. And the other 50% is phospholipids. Okay, does that make sense? Mm-hmm. Now, so this is the model that came about in the 1970s. And pretty much this is the model that people stick with. And the reason I say that is the conception of what the membrane was made, how it was made, was changing over the years. And it took a lot of electron microscope pictures to kind of get it firm in their mind. And then now they can label, like they can label some of the proteins. But the kind of thing where they found the proteins can move within a membrane, where they found that out was labeling some of vertebrates have lymphocytes. And the lymphocytes is a type of white blood cell that fights off bad guys, okay? But certain kinds have receptors on them, and they found that they could label these receptors with fluorescent types of molecules, and if they just label one of those lymphocytes with the receptors, Yeah, the receptors are all pretty much evenly distributed all around the cell membrane. A number of things are three-dimensional. We're looking at two-dimensional, but remember, this the cell is three-dimensional, so the cell membrane here would be coming out like this and coming, every way.
Yeah.
But anyway, so if you think of a sphere, maybe the lymphocyte's like a sphere. These receptors are all over the thing. If that lymphocyte came in contact with something that it can respond to, it could hook onto its receptors, the receptors would all move up to one part of the cell, which they call the pole, like the North Pole or something that they call it the pole. They just call it that. But they'd all move up there. And so they go, oh my God, these proteins, these receptor proteins, were actually able to move within this cell membrane. So that's when they started using the term fluid. People who saw this said it appears that some of the proteins, not necessarily all of them, but some of the proteins can move within the cell membrane. So let's write this down. Now we're going to talk about the fluid mosaic model, okay, of the membrane. Fluid means, I'll say fluid because many proteins, many membrane proteins, if you put it that way, many membrane proteins can move within the membrane. So that's what the idea of the fluid part. Mosaic,
Many membranes can move within?
they're moving within the membrane. I mean, what I was many membrane proteins. Sorry, thank you. Come on, see what don't put this on YouTube. Many membrane proteins. Sorry. So what happens when you try to talk too fast? Okay, it's called the fluid part means it's called that because many membrane proteins can move within the membrane. Okay, it's called mosaic. Mosaic because the proteins that are in the cell membrane will be different depending upon the cell. And even within your own body, the membrane proteins in a liver cell will be different from the membrane proteins on a nerve cell, will be different from a kidney cell, et cetera, et cetera, et cetera. So mosaic because the membrane proteins, I mean, they may share a lot of the same proteins, but still there'll be all kinds that are different. Mosaic because the membrane proteins differ from cell to cell. Okay, so this is pretty much a model. I don't think it's going to change much because it pretty much encompasses just about everything here. But from my perspective, what I want you to see is that the membrane is composed of about 50% of these phospholipids, proteins, which are much more in much more quantity than I'm indicating in my drawing. And that some of the proteins, go all the way through, some don't. A lot of these proteins are helping to provide structure, but others have other functions, and I'll talk about those in a minute. So the fluid mosaic model, why fluid again? Why fluid? Because some of these proteins can move within the membrane. And mosaic, because from one cell to another, the proteins are different. They used to think it was that all membranes are pretty much alike, just a bunch of phospholipids, a bunch of proteins. But it's not that. It's much more specific. Depending upon the cell, different set of proteins. Even if you look at a cell, let's just say, if I drew a little simple picture here, and I said, okay, so here's a blood vessel, and here's some cells. Okay, so here's a cell here, here's a cell here, here's a cell here, okay. If you look at the membrane proteins, now there's a little tiny, look big in my picture because I made it, with a little tiny bit of fluid right here. But if you look at the membrane proteins here, versus the membrane proteins where the cells come in contact with another cell, versus the membrane proteins up here, there'd be a different complement of proteins in each of those locations. So even within a cell, depending upon what that cell membrane's facing, is it facing another cell? Is it facing fluid near the blood vessel or whatever, it's going to be different. So keep in mind the mosaic, that's an important aspect of that model. The proteins are different. So what? Well, let's talk about the so what. Let's talk about the function of membrane proteins, because this is critical here, that you realize. Now, so far, what have we said can get through the cell membrane? We said lipid-type materials and water, probably, right? But yet, cells have to get nutrients. Oh, by the way, maybe the gases can get through. Gases, CO2 and oxygen get through too.
Even smaller than
really
water,
tiny.
yeah.
Say it again.
Yeah, smaller than water.
Yeah, they're
hydrogen.
smaller than
You
water,
can't even
exactly.
keep hydrogen in a lot of stuff; it'll get out.
But what about things that the cell's going to need? It's going to need sugar. It's going to need amino acids to make proteins. It's going to need all kinds of things. How are those things going to get in? Those things like water, they're not going to go through. So, but it turns out, or ions. You need sodium ions, and maybe potassium ions, chloride, and maybe calcium. How are those things going to get through? Well, okay. So let's look at what I titled there: the function of membrane proteins. One other thing, by the way, in terms of the test, if it's on the study guide but I forgot to talk about it, or I didn't get to it, it won't be on the test either. Only if I talk about it and it's on, and then I put it in the study guide. In case I forgot to put something in the study guide that I did talk about, and I remember that, I'll tell you before the test. Not the right time to take the test, but the time to do the study.
By the way, the answer to this one is.
Yeah, right. Don't forget. The answer is yeah, right. Okay, so let's talk about the function of membrane proteins. And then this will help answer things like how can other molecules get in. I like to put it in 5 categories. You might find it 6 categories, 7 categories. Some people put it in 4 categories. I like 5 categories, because it depends upon how you put things, how you put them in categories. And just an aside here, having to do with biologists and putting things in groups. There's always the conflict, not conflict, let's just say the disparity between the lumpers and the splitters. The lumpers, when you're categorizing things, the lumpers want to lump everything in the fewest number of categories possible. And the splitters like to separate them into, so if there's one slight difference, they'll make them different. And so, as an example, just I'll give you an example. In microbiology, there's what I call the blue Bible of microbiology. It's actually a whole bunch of volumes, but it's called Burgie's Manual, and it's blue. But anyway, the edition that came out about 25 years ago or so, if you look up Streptococcus, Staphylococcus is the one I was going to talk about. Staphylococcus is a type of bacterium that can cause some pretty nasty infections. But anyway, if you look up Staphylococcus, there were 3 species. The next edition had, I think it was 19 species. So whoever was in charge originally of getting the information and compiling it and coming up with decisions about, how you differentiate different species of it, they said, well, let's just kind of lump these things in and make 3 species. The next time they had more information, and the people in charge were more the splitter types, and if there was just a little bit of difference between this guy and this guy, they say, let's call them a different species. It's just it's a decision someone makes, arbitrary type decision. The newest edition, which is still in the process of being formed, I don't know how many species there is, but my guess is probably splitters are still in fact in force, I should say. Whatever. So I like to put 5. I'm sort of semi-splitter, but not a total splitter. I'm not a total lumper in this case. Function of membrane proteins. I'm going to write it down, but I want you to leave a little tiny bit of space between them. So, channels. I don't want to talk about each of these, but I just want to write them down. Channels, pumps, receptors. In my physio class, receptors! That's the answer to a lot of questions I ask in class. So, not on test necessarily, but when I say, how do you think such and such could happen? At least half the time the answer is receptors! So that becomes the key word in my physio class. It won't be the key word in this class, but it'd be a nice word to have. And of course, if you say receptors, you don't just say receptors, it's receptors! Just I have to tell you. Receptors, let's see, what are we doing now? Pumps or genital receptors. Structure. Let's do enzymes next, I guess. Enzymes. I don't really talk about enzymes, but keep in mind we will. Structure. Okay. So, I was just saying, I was posing the question, how do things get in? Things like amino acids or sodium ions and all that. Yeah.
Receptors.
Oh, no, but it was a good try.
Through the protein channel?
Yeah, well, so here's the deal. There are some of these membrane proteins that go all the way through, and think of them as just a tube.
Tunnel, yeah.
It's some kind of a protein tube, and that's what we call it, a channel, that goes all the way through. Okay, so so let me just say, but think of it as a tube that goes all the way through, and certain things can get in or out, you're able to get in and out. So let's just say it's a tube, I'll say tube, in quotation marks, a tube, so it's got an opening in it, through the membrane. It's got certain characteristics, highly specific. Now this is an important idea in biology, specificity. Highly specific. What that means is certain things can get through a specific channel, but nothing else can. For example, sodium ion. There are certain channels that only sodium ions can go through, but nothing else can. There are certain channels that only potassium ions can go through, but only potassium ions. And yet, if you look in the periodic chart, sodium and potassium are very similar to each other. In fact, if you were trying to separate them from each other, it's kind of tough. You can do it. They're chemical means of separating, but they react so similar to each other that they're hard to separate. And your cells can do it. Your cells can separate sodium ions from potassium ions, or they can let sodium ions in but not potassium, and vice versa. So they're highly specific, but also passive. Passive. That passive means that they're just always there. They're just always, but no work is involved in having something go through it. It'd be like a window. If you just open the window, what can come through it? Air can come through it. Maybe small birds can come through it. Dirt and dust can come through it. You wouldn't be able to drive your car through the window, right? So it is keeping certain things out and letting certain things in. But it's passive. The window isn't doing any work. The window is, as I said before, isn't grabbing you and pulling you in or something like that. It's passive, no energy. So passive means no energy is used. No energy is used in the working of the channel. And because it's passive, things can only move by normal laws of diffusion. Next week, I think we're going to do a lab in diffusion and osmosis, okay? But when things are diffusing, I'll just say it now, and if I have time, I'll come back to it. When you're talking about diffusion, you're talking about molecules moving from where they're more concentrated to where they're less concentrated. They never go the other way, unless you put energy into the system. In other words, you have to use it. The next thing, pump. If you, in other words, it's like water. The water doesn't go uphill, right? Unless you pump it uphill, but it won't go uphill by itself. Well, in the same way that when you were talking about passive, we're talking about just normal diffusion. Things will diffuse from where you have more of them to where you have less of them. And you guys, most of you aren't in my lab, so it will be explained pharmacokinetics.
You have a lab?
My lab. I mean, I'm teaching by one lab, but from my other lab lecture section.
Oh.
But I'm saying you're not in my lab section, so you won't get my description. But what I usually try to get when I talk about the laws of diffusion, I say things go from where they're more concentrated to where they're less concentrated. Why? How do they get there? First of all, how do they move? And they're not taking the bus, right? When you're talking about diffusion, they're not taking the bus. They move. Here's the deal: the universe runs on statistics. Yeah, there's the molecules are in motion, so there's kinetic energy. Of course, if you raise the temperature, they're in faster motion, right? And so things are moving around. But the universe runs on statistics. And so, and if you're talking huge numbers of things, you can make judgments about, what's going to happen. And if you have a whole bunch of things right here, and they're bouncing around, they're going to bounce this way and bounce away from each other. And so I actually did this experiment. By the way, I've got to tell you something about diffusion. But let the question go ahead.
That was a question of the statement.
Go ahead.
That was it. On either side of the cell membrane, the number of molecules,
Right.
whenever it's transosmosing, have to be equal because of how it operates.
Exactly. And we'll come back to this idea. So, but let me just say, I did an experiment which is analogous to diffusion. Diffusion, as it turns out, is incredibly slow. I mean, incredibly slow. And most biologists aren't too good with the concept of diffusion, so you need the physical science people to try to help explain it. So most of the examples they give of diffusion in biology or in textbooks of biology are not. The amount of diffusion is minimal, and mostly what they're talking about is convection. But it's a good analogy of diffusion. So here's a good analogy I used once in a lab class. I took a bottle of ammonia, I set it right in the very back of the lab. I didn't say anything to anybody, I just let it be there. I opened up the bottle, and so they're sitting there, and all of a sudden, so the ammonia's coming out of the bottle and kind of diffusing around, mostly by convection, it's moving, and then suddenly the people in the very back row say, "Oh God, what's this terrible smell?" And everyone else is, "What terrible smell? We couldn't smell anything." And a few minutes later, the next lab, "Oh God, we can smell it too. What is that?" Anyway, I had to sort of stop the experiment because people are about to riot. But what was happening is you had a lot of ammonia in that container. When I took the top, and the molecules are moving on, but they're starting to bounce around, and they're bouncing out and all that. And if we waited long enough, they'd be more or less evenly distributed in the room. Okay, here's where I'm talking about statistics where it comes into play here. Think of it this way. What if we have the molecules evenly distributed in the room, and you took an open bottle, empty bottle, it had air in it, but no ammonia, and you put it in the middle of the room? What are the chances of those ammonia molecules ending up in that bottle? Way less than your chances of winning the lottery. How many of you won like millions of dollars in lotto? That's what I thought. Darn it, I was hoping one of you did because I had this beautiful bridge that was for sale, Anyway, but here's the thing, the reason they could offer millions of dollars in prizes in the lotto is because the chance of winning the lotto was so slim. I don't know what the present lotto, I don't know what the chance is like, maybe 1 out of 57 million. Depends upon how it's set up, and I don't really know how it's set up. And here's the deal. In terms of statistics, your chances of winning the lotto are almost exactly the same whether you enter or not. And that's a weird concept. When you think of it that way, you may not be so inclined to enter. Your chances of winning it if you don't enter it are absolutely zero, right? If you enter it, very close to zero. It's like 0.00000 something. I don't know how many zeros it is because I don't know what the percentage is. But my point is the chance of winning it is slim. And you might say, well, someone's got to win it. Well, no one has to win it. But if several million people enter it, then there's the more the people enter, they're picking random numbers, the greater the chance is that someone's going to win it. There's no magic that they have to because it could go on weeks and weeks and weeks and weeks and weeks and not anyone win it. Whatever. So back to statistics. So the universe runs on statistics. So what I'm saying is that if you just took something that's real concentrated like that ammonia and took the top off, just randomly these molecules are going to bounce around, and they'll just bounce around, so that pretty soon they're, as I say, roughly evenly distributed. But there's no way the chances of them all coming in to, from being evenly distributed and come in there is not going to happen unless you put some energy into the system, unless you pump them in. If you have some kind of an ammonia pump that can grab the molecules and pump them into the jar, okay, then it can happen. You know what I'm saying? But you'd have to put some energy into the system. So these are passive and highly specific. Things move in or out depending on the laws of diffusion. So molecules go in or out depending on laws of diffusion. And as I say, you'll talk about this. this later in lecture lab. As I said, the problem with diffusion is that it is such a slow process, and yet, as I say, biologists didn't use convection as diffusion. It's not right. It's really diffusion. For an oxygen molecule to diffuse between my thumb and my finger, if it was pure diffusion, it would take about 10 hours. That's how slow diffusion is. But of course, air is moving around. There's all these convection currents, and that moves things around fast. In your body, to move things around, we have a heart that pumps the blood. But once the blood goes into the capillaries and gets to, near the cells, things diffuse out of the capillaries, and then by diffusion move towards the cells. But the space is so tiny. The distance that they have to move is so tiny. That's good. That's a good thing. So that diffusion works. Anyway, but they have to use the laws of diffusion. So as I said, we have various kinds of channels for sodium and potassium and calcium and stuff like that. But they're highly specific. If it's a sodium channel, only sodium can get through. But it's only going to follow the laws of diffusion. By the way, some channels are always open. Some channels are open sometimes and closed other times. We call those gated channels. Gated meaning, you close the gate, you open the gate. So you don't need to know this for this class, but for my physio class, if you ever take my physio class or a physio class, you need to know that some channels are gated. They'll open at certain times and be closed at other times. But other channels are always open. All right, but so I was going to say, so sodium. Let's take sodium or potassium. Those molecules are relatively evenly distributed in your body. Okay, sodium. Let's just say take sodium. What about iodine? Iodine. We need small amounts of iodine to make thyroid hormone. Okay. All right. We get iodine from plants, and if the plants get it from the soil, if you live near the ocean, you're going to have plenty of iodine, so it's not a problem. If you're worried about not having enough iodine, you can get salt that is labeled iodized. That just means they put a little bit of iodine. I think they put something like 1 part per 200 of potassium iodide, and the rest is, sort of table salt. You don't need to worry about having enough iodine living in Humboldt County. You definitely don't have enough. So it's not likely you're low on it. But anyway, back to the iodine. So we need iodine. We need a certain amount of iodine. If it was like sodium, you'd expect it to be pretty much evenly distributed in your body. No, 99 point whatever percent of it ends up in your thyroid. Well, it didn't get there by diffusion, right? It didn't get there by diffusion because that's just going to follow the laws of diffusion. By passive, you didn't get through just passive channels because that would follow the laws of diffusion. You expect to be all over. So it had to get pushed in there, had to get pumped in there, and that's the idea of a pump. So this is like a tube. Again, I'm going to call it a tube through the membrane. It's also highly specific. But active, meaning that energy is used for it to work. Now, energy in the form of ATP, we haven't really talked about ATP, but a lot of you have heard about it, and we will be talking about it in the future. So energy used to move things through whatever kind of pump it is. And it could be moving things in, could be moving things out. Again, it's not, depends on what we're talking about. And the energy is usually in the form of ATP. But we'll talk about ATP in the future. So again, it's highly specific. Each pump will only pump certain things, but again, energy is used. So to get all that iodine into your thyroid gland, there's some very powerful iodine pumps in the cell membranes of thyroid cells. Not in other cells. That's what I mean about that fluid mosaic. Not every cell has an iodine pump, just thyroid cells have. Anyway, okay, so yeah.
That removing iodide, that's what the whole point is. Okay, removing iodide from your system slows down the production of
Thyroid
thyroxine.
Yeah, you don't want it. You don't want it.
I do, but
Oh, you might, but most of us don't want it because you need thyroid hormone to have your metabolism going.
or you gain weight.
And if you have too much thyroid hormone, then you get hypothyroid. You get all. And I may be a bit hypo, but I'm not hypothyroid. But you get really hypo, and if your thyroid level goes down, you start getting. So you want to be right in the middle. So thyroid hormone. Turns out thyroid hormone is weird. It's such an important hormone, and yet it's not absolutely essential for life. Meaning, if you run out of it, you don't die. But you go into what I remember as a kid listening to some of those science fiction programs, and people get into suspended animation, and I didn't know what that was in those days. I still am not sure, but I know what they're trying to say. And if you saw Star Wars movies, what was it?
Carbonite.
Han Solo was carbonized or something like that.
Carbonite.
Yeah, he was carbonite.
Frozen in carbonite.
Frozen in carbonite. Well, that would be like suspended animation where nothing's happening. Your metabolism. Well, that's what happens if your thyroid hormone stopped being made. You would be incredibly slow. Your metabolism would slow down, your temperature would go down. Anyway, it doesn't happen usually, but I mean, if it did happen, you'd be as close as you could get to suspended animation.
Yeah.
Anyway, okay, receptors. Oh, sorry, I get excited about receptors. Receptors are highly specific. Again, and they bind. They bind to something. It depends upon what kind of cell, what kind of receptor. But we'll say bind to something, but in this highly specific way. For example, I was mentioning hormones. Well, we have all kinds of hormones that our body makes, and various cells make receptors for these hormones. And if you have a receptor to the hormone, you'll respond to the hormone. If you don't have a certain cell doesn't have a receptor to the hormone, it won't respond. That's fine. That makes sense. But we also have receptors on I mentioned lymphocytes earlier too. And lymphocytes have all these receptors on them so that they can respond to bad guys. And if they come in contact with a bad guy, then they can latch onto them and maybe make antibodies or whatever they're going to do, depending on what we're talking about. So there are all kinds of receptors. Cells have receptors, when a cell is growing and dividing, if they come in contact with each other, they usually stop growing. And that's called contact inhibition. It has to do with various kinds of receptors. One of the things that goes wrong in cancer is they lose this contact inhibition and they keep growing. They come in contact, they don't care, they just keep growing, and they form a mass. We call it tumor. That's not a good one. Of course, it can spread. That's even worse. Yeah.
I was going to ask about people that get organ transplants.
Yeah.
So the medication that you put on, is that supposed to stop the receptors from working, or just stop the forming?
Well, it's cutting out the entire. They don't want to cut the entire. Okay, that's a good question. So here's the thing. We all make receptors, but there's certain receptors that are, that we all share, and pretty much, and we share a lot of these receptors with our closest relatives, gorillas and chimpanzees and all that. Chimps are our closest relatives. So a lot of receptors we share, they're the same. But then we all have a group of receptors that are slightly different. And that's what makes each of us slightly different from the other. I mean, that's not the only thing that makes us slightly different, but in terms of cells, that's what makes the cells slightly different, these receptors. If you have an identical twin, you could have an organ transplant, that'd be no problem because, your identical twin makes exact same receptors as you do. Exact same membrane proteins. I'm really talking here about membrane proteins, sorry, in general. You make the exact same membrane proteins on your cells, so you could have a transplant, it'd be fine. But if you try to give a transplant to someone else, or let's say you needed a kidney transplant or a heart transplant, your immune system is going to respond to all these membrane proteins and it's going to mount an attack and try to get rid of this foreign body. So what they try to do is inhibit that immune response, so that you're not rejecting this foreign transplant, so that you can have this and survive. But they can't totally stop it because if you're inhibiting your immune response, then you're more subject to getting infections on. So there's a lot of things to consider. It's not just a simple idea, but that's sort of the basic idea. Yeah.
So one thing you document is when your body starts to receptors.
Well, I don't know. Do they ever take you off? It depends upon what the transplant is. I don't think they ever I don't think they ever take you off, do they? I think that, yeah. And by the way, something that can be really tough is bone marrow. Every so often someone gets a bone marrow transplant. And the bone marrow is where all these red and white cells are made, especially the whites, which are the immune response cells.
Yeah.
And what can happen with wasn't there a movie, the movie about the boy in the bubble? It's a real story, except that in the movie,
Leukemia.
who was it that played in the movie?
John Travolta.
Yeah, Travolta. Oh, yeah. I didn't see the movie. I just heard about it, but I refused to see it. But,
Yeah.
but the case was based on a real thing,
Mhm.
and this boy didn't make these. He didn't make certain immune response cells,
Mhm.
and so they knew about it because he had two siblings that were born before and died very early of infection, and he lived down near the NASA or whatever, and so they actually tried an experiment where he lived in this bubble where everything he got, air, food, everything was totally sterile, so it wouldn't introduce any microorganisms, and to see if they could keep him alive to maybe get a good one, maybe a bone marrow transplant. And so I think he got up to about age 8 or 9. I don't know how old Travolta was in the movie, but well, older than the kid.
Yeah.
But he got up to 8, maybe as high as 12. But here he lived inside this bubble. His mom couldn't hold him. She could hold him through this plastic thing. I mean, it's not a normal life. And the only reason they did it was an experiment, But at one point, and I think he was around 12, they were looking around the world for tissue transplant people and to try to get the closest match they can. They couldn't get it exactly, but if they could get it close enough and then inhibit the response, they could maybe get it to work. And it does happen for people now. This was a number of years ago. In his case, they found something that was close. They tried it, and unfortunately, it wasn't close enough, and the transplant rejected his body. And that sounds weird, but it's the transplant, which is bone marrow. That's what makes the cells that do the rejection, so it rejected his body, which meant that he died. I think in the movie, didn't he walk out of the bubble?
Yeah.
You
Yes.
thought it was going to be happily ever after, right? But it's not that simple. Well, Travolta, he probably did live happily ever after. I don't know, but yeah.
No, he's a Scientologist.
Louder, I'm sorry.
What about blood transfusion?
Yeah, so blood transfusion, they just try to match it as close as they can. And main things are those A and B things, which I'll explain to you someday.
They don't give people like medicine.
No, not for blood transfusion.
Because the body will just
The red cells have certain membrane proteins, and if you could match some of them, and the main one, the match of the A, B, what they call the ABO protein. And actually, we're going to do an experiment at the end of the semester. We'll maybe go over that a little bit. Let me go on with this real quick. I want to finish this though. Okay, so receptors. These are very, very important little guys. Enzymes. Now, we haven't talked about enzymes, but enzymes are biological catalysts, and I'll say more about those in the future. Because they're very important. They catalyze reactions. They allow chemical reactions to work. And I will be talking about this, but I'm going to wait on it. So just keep it that. Structure. You know what structure means. Just to help hold things together. We talked last time about the cytoskeleton. Okay? So the cytoskeleton is this mostly protein kinds of things. Other things involved too, but mostly proteins inside the cell. So if you have a cell, like this, you have all these proteins in here. You have some molecules that can kind of hook together various proteins, and so it helps to provide some structure to the cell. We're not going to get into that kind of detail. You don't need to worry. In general, just as a generality, any one protein will have one of these functions or more than one. It could have, something could be, and in many cases there might be a channel and a receptor at the same time, or whatever. So things can have, or they can provide some structure too. So things can have more than one function. So in other words, so here's the point. All these membrane proteins, they're there. They're not just there as some passive thing. They're an active part of the cell. It makes the cell membrane an active part of the cell. And it's these channels and pumps and receptors and enzymes that determine what's getting in the cell, what's not getting in, what's getting out, what's not getting out, and therefore it's going to have a big effect on what the cell does.
So these pumps are kind of things that would grab somebody through.
Yes, grab you and pull you inside. Yes, exactly, or throw you out of it. Yeah, another question? No? Okay, let's talk about these organelles briefly. Oh, I'm sorry. What? Go ahead.
Structure. You just assumed.
I just said.
We just know what it is.
That cytoskeleton, instruction of the cell membrane. I didn't want to go into any detail, just to help provide shape, structure, whatever, integrity to the cell membrane. Would you like that integrity? That's really okay, huh? Let me say something about organelles. So, and I mentioned this the other day, but when they looked at cells in the microscope, once they started looking at them, they started staining, getting different stains to stain things. They noticed that in eukaryotic cells, there are all kinds of structures that you can see. With different stains, you can see different things. And since the body is made of organs, remember I told you this, then they saw, hey, within cells are what look like little tiny organs within a cell, and so they call them organelles.
Yeah.
I told you I had a student that kept trying to call them organelles. I said, no, they're not organelles, they're organelles. But anyway, whatever. So, they're just little membranous structures. Well, not always. All cells have ribosomes. These are really tiny. They're not membrane bound. Ribosomes, I'll just write this down here. Ribosomes, not membrane bound. Not membrane bound. No membrane around. And anyone know what ribosomes are for?
Ribosomes make proteins.
Protein synthesis, yeah, they're for protein synthesis. We'll be talking about these in the future, but yeah, for protein synthesis. And so all cells have this, including bacteria, a little different shape and size, but they still have them. But then only eukaryotic cells have membrane-bound organelles. Now, what was the big deal about this membrane? I said it's a barrier, right?
Mm-hmm.
So it keeps the outside out and inside in. So when you're looking at these membrane-bound organelles, it keeps the outside of the cytoplasm away from what's inside, and the inside away from the cytoplasm. So it's sort of like a house, a little. Well, let me tell you this. First of all, let me say, I'll put this little arrow here in all cells. I had it over there, but I want to put it here too. So ribosomes are in all cells. Only eukaryotic cells have the membrane-bound organelles. So bacteria, no membrane-bound organelles. All right. Let's just write this up over here. Prokaryotic, no membrane. I'm repeating myself, but on purpose because it's important. No membrane-bound organelles. So, kind of a simple comparison would be like a one-room cabin that you might live in versus a three-bedroom house. Well, let's say a 6, 7-room house, whatever it want to be. In a one-bedroom cabin, you'd have a little area where you cook, another area where you do your bathroom functions, another area where you sleep. It's all within the same room, right? I mean, everything's right there in the same room. But you have all the normal functions you have for a house. What if you have a big house? You have a separate kitchen, a separate dining room, maybe a separate bathroom, separate bedroom, separate family room. You have a separate room for each of these functions. So you do all the same things that you in a big house compared to a little house, compared to one-room cabin. It's just that in the big house you have a separate room for each of these things. That's really what it's going to amount to with what's happening in prokaryotic cells that don't have these membrane-bound organelles versus eukaryotic cells that have membrane-bound organelles. Okay, they still have the same functions. A membrane-bound organelle over there. They still have the same functions. So anything that the eukaryotic cell does, the prokaryotic cell can do also, and vice versa. In most respects here, but it's just that they're not in separate compartments. It's just a way to compartmentalize these membrane-bound organelles, compartmentalize various functions. We'll take some examples, but go ahead, question first.
So you're saying our cells are basically like luxury homes. Is there any reason for the membrane, or is it just because?
Well, it does allow these cells to get bigger and more complex. These guys, and so these are much bigger cells, more complex, and maybe they can do more things. I mean, same thing, but more of it. You know what I'm saying? So they're more elaborate. So examples of the organelle. Examples of organelles. Nucleus. Let's just write some of these down. Mitochondria. Endoplasmic reticulum. Oh, that's a long one. It's usually just called ER to get it so much easier. Endoplasmic reticulum, Golgi body or Golgi apparatus. Lysosomes. In plants, the plants have all the same thing. Chloroplasts. Actually, plants and algae and other things, but let's just say plants. But plants have everything that the animal cells have, plus the chloroplasts. But let's just real quickly, what's a nucleus? It has a DNA. It has more than just DNA, but in particular it has a DNA. We'll be talking about that. That's the genetic information. It tells how to, the blueprints that show how to run the cell. Mitochondria, where most of the ATP is made. ATP is our energy molecule. We'll be talking about this in the future. ER, endoplasmic reticulum. These are tubes, channels, whatever.
Some are rough, some are soft.
I'm sorry, say it again.
Some are rough, some are soft.
Say it one more time.
Some are rough and some are soft.
Oh, yeah. Oh, okay. Rough and smooth is what we usually call it. Rough and smooth. But yeah. But I wasn't going to get to that. But tubes or channels. That sounds like, it sounds so much like membrane protein. These are sheets. Let's say sheets or tubes. But membranous sheets or tubes. Things happen in there like processing of proteins. Other things happen. I'll just say processing. Without going into any detail, maybe we'll have time to talk about sometime. Processing of proteins, etc. The liver has a lot of this. More processing of proteins. Golgi body. I didn't even, not every protein, but I'm not getting into this right now. Lysosomes, oh my God, they sometimes call these suicide bags. I don't write that. But they contain digestive enzymes. So cells that eat a bacteria, these digestive enzymes can kill the bacteria. So I'll just say contain digestive enzymes. I'll tell you more about those sometimes. There's so much I could tell you about, and I don't have time. And then chloroplasts, of course, for photosynthesis. So here's the deal, just real quickly before you go. Bacteria don't have organelles. So bacteria don't have a nucleus. Do they have DNA? Absolutely. And it's still their hereditary information tells them what to do. They don't have mitochondria. Do they make ATP? Of course. They just don't make it inside a little compartment. Do they process proteins? Absolutely. They just don't process it in a special compartment. Do they have digestive enzymes? Do some of them photosynthesize? Absolutely. Does that make sense in what I'm saying about this? So,
Yep.
So you just get that basic idea that in a eukaryotic cell, they tend to be bigger cells and they have little compartments where they do things. Little separate compartments, like, separate rooms in a house.
All right.
It's all just right there. Yes, just like a one room house. And then, they have the DNA.
Stop, baby, stop.